This second video will cover the Krebs cycle, which occurs after glycolysis and the oxidation of pyruvate. We'll start by reviewing some of the key players. All of the carbon-based compounds will be colored in orange. ATP and ADP I'm using pink for. The electron carriers I'm using light blue for. Oxygen I'm using green for. And water I'll use dark blue for. So when we left off, we had shuttled pyruvate in through transport proteins into the mitochondria, into the mitochondrial matrix, and that pyruvate had been oxidized into acetyl. This step right here is the step between glycolysis and the Krebs cycle. Okay, and this step actually has a name. This step is called the oxidation of pyruvate. So maybe we'll call this step like 1B. It's between glycolysis and the Krebs cycle. This step is called the oxidation of pyruvate. And like I said in the last video, we have this pyruvate, which is made of carbon, hydrogen, and oxygen. And we're going to cut off one of the carbons to produce acetyl. When the carbon comes off, okay, electrons are actually captured by NAD, the electron carrier, to make NADH. Okay. Now, after the oxidation of pyruvate, we have the Krebs cycle right here. The Krebs cycle happens inside the mitochondria and the matrix of the mitochondria. So just a quick note about the matrix of the mitochondria. And a quick reminder of form and function. So the form or structure of the matrix of the mitochondria is fluid containing, we know the matrix of the mitochondria has its own DNA and its own ribosomes, but more importantly, for cellular respiration, it also contains enzymes that can help with the Krebs cycle, gases like oxygen and carbon dioxide, water. It also contains pyruvate and acetyl, and many other sugars that are involved in the Krebs cycle. And finally, many electron carriers. And by electron carriers, remember we mean NAD+, which can be converted to NADH, FAD+, which can be converted to FADH2. So this structure, this form of the matrix of the mitochondria, okay, all the things that are inside the matrix are key for the function, which is reactions in cellular respiration, right? The oxidation of pyruvate, the Krebs cycle, right? Helping provide things to the electron transport chain for electron transport chain and oxidative phosphorylation. So to continue the process of cellular respiration, that's the function. And luckily that can happen because of all these specialized things that are found in the matrix. So when we're talking about the Krebs cycle, okay, we're talking about a series of chemical reactions, okay, that occur because of all these enzymes and all these other necessary fluids, okay? So for the Krebs cycle, and I'm going to write that one more time here, we're talking about acetyl, which is a 2-carbon sugar, and it's going to join with the help of an enzyme to another 4-carbon sugar. That 4-carbon sugar is called OAA. And it's just constantly recycled in this process. So it's constantly found in the matrix of the mitochondria. Okay. So when OAA and acetyl join together, they form the first compound in the Krebs cycle, which is a 6-carbon sugar called citric acid. So sometimes the Krebs cycle is also called the citric acid cycle because this is the first kind of major sugar that is produced in the Krebs cycle. Luckily, you don't need to know all the names of the different sugars found in the Krebs cycle. All you need to know is that Acetyl joins with another sugar, a 4-carbon sugar, to make citric acid, and then slowly this citric acid is broken down into OAA again through a series of enzyme catalyzed reactions. As citric acid is broken down again, the energy in these bonds is released, and it's used to make more NADH and more FADH and a little bit of ATP. So during this process of one turn of the Krebs cycle, if you put one acetyl in, you make 3 NADH, 1 FADH2, and you even make 1 ATP. As these carbons are breaking off, so that citric acid can be converted back into OAA, they leave as a waste product, so we also produce 2 CO2 here. So remember, in glycolysis, we produce 2 pyruvate. 2 pyruvate go through the oxidation of pyruvate, but only 1 acetyl enters the Krebs cycle at a time. So the first acetyl will enter the Krebs cycle, join with an OAA. This cycle will continue to produce carbon dioxide, NADH, FADH, and ATP. Then the second acetyl will enter the Krebs cycle and do the whole thing. So if you're talking about processing an entire molecule of glucose, you'll produce 4 CO2, 6 NADH, 2 FADH2, and 2 ATP. So the purpose of the Krebs cycle really is to continue to get as much energy out of this acetyl as we can. Okay, and this is all happening, like I said, in the matrix of the mitochondria. Okay, so what is the purpose of all of this? The purpose of all of this is to make as many of these electron carriers as we possibly can. So before we moved into the last part of cellular respiration, which will be in a third video, I want to go back and explore this first equation again. Okay, here's our glucose. We used our glucose during glycolysis. In glycolysis, this glucose was broken down into pyruvate. In the oxidation of pyruvate, the glucose was broken down into acetyl. And in the Krebs cycle, we get the last bits of energy out of that acetyl by cycling through citric acid and a bunch of other sugars as well. We haven't used our oxygen yet, but it's coming up in the last video. We've produced a lot of carbon dioxide. We produced carbon dioxide, not in glycolysis, because in glycolysis, we neatly split the glucose into pyruvate, but we produced carbon dioxide in the oxidation of pyruvate and in the Krebs cycle. We haven't produced a lot of water yet, but we're coming to that. What about ATP? We produced ATP in glycolysis. We didn't produce any ATP in the oxidation of pyruvate, but we produced a tiny bit of ATP in the Krebs cycle. Most of our ATP is going to come from this last step of cellular respiration, the electron transport chain and chemiosmosis, together called oxidative phosphorylation. Again, that will be in the last video. So that's kind of the purpose of the Krebs cycle, is to finally break down the sugar as much as we can and produce all these electron carriers. Okay, so why do we need these electron carriers? I want to leave us with kind of the flow of electrons. Okay, so when we think about electron flow in this process, we need to think about where the electrons are coming from. We have high energy electrons from glucose. And really, that's the point of glycolysis, the oxidation of pyruvate, and the Krebs cycle, is to transfer those to electron carriers so that we go from NAD plus to NADH, FAD plus to FADH2. So we're transferring these electrons to electron carriers. They're literally going to carry the electrons to the next step, which is the electron transport chain. And then finally, after the electron transport chain, the low energy electrons will be transferred to oxygen to form water. We haven't gotten to that step yet, but this is really the meat of what we're talking about in glycolysis, the oxidation of pyruvate, and the Krebs cycle.